absolutely amplified. Can you hear me? Good. How do we adjust the volume? 1, 2. 1, 2, 3. Can you hear me? Oh, you're the volume.
It's okay. So let me introduce Professor Mark Solms, which is one of the leading person of neuropsychoanalysis, with what is an interdisciplinary field between the neurosciences and psychoanalysis. So here it is.
Thank you. Thank you very much. It's wonderful to be in this beautiful city and in this beautiful room. As you no doubt assumed, I'm going to be speaking in English. I'll try to speak as slowly and clearly as I can. Wow, it really is. Now, is it too loud now? As you've just heard, the field that I'm most involved with is called neuropsychoanalysis. And what I'm going to do with tonight's talk is try to illustrate what that field is all about. I'm going to do that with reference to one particular topic, namely the brain mechanisms of dreaming. And I'm going to be addressing a specific question, which is the title of my talk: why do we dream? I will go into all sorts of little nooks and crannies as I go along, mainly telling you a story, telling you the story of how my own research in this area of brain mechanisms of dreaming, how it unfolded. And I will end with the results, or the preliminary results, of a study that I'm busy doing right now. But I must emphasize that I'm using this story and these bits of science that I'm going to refer to as we go along. I'm using it in order to illustrate what this whole field is about. And so that explains why I'm taking so many little turns as I go along. There are many subplots to the story that I'm wanting to tell you. Well, let me start with the obvious. When we ask the question, why do we dream? This is a question that in a way has been asked since time immemorial. People have evidently always been interested in dreams. They seem always to have believed that dreams mean something, that dreams are important, that dreams are worth paying attention to. If you look through all the different cultures and all the different times that dreams have been taken seriously in this way by our species, the common denominator seems to be something along the lines that the dream communicates something, it conveys something from another place. It might be from God, it might be from the future, it might be from the spirit world, the ancestors. But that's sort of the gist of the idea, that there's some hidden meaning in dreams that come to you in some indirect way from some place that you don't normally have access to. When scientific views of dreaming began to establish themselves, they very much took the opposite view. Without there being any particular evidence one way or the other, the view that gradually came to dominate in the 18th and certainly in the 19th century was that dreams are nonsense, that dreams are the products of a sleeping brain. Therefore, they're the products of a brain that's not working properly. It's asleep. So it makes kind of semi-random images, thoughts, experiences, because that's what brains do, and brains that are asleep don't do it properly. The image of a drunk pianist sort of running their fingers meaninglessly across the keyboard was the image that was used to convey the relationship of dreaming thought to waking thought. It's like that drunken plim-plom nonsense compared to proper music. Traume sind Schaume, dreams of froth, was the view that dominated at the time that this man on the screen, who's going to feature quite a bit in my story, when he came onto the scene, roughly around the time that that photograph was taken, he was beginning to develop the opposite view to his scientific colleagues, and was reverting to this old folk psychological notion that dreams carry meaning, and what's more, that the meaning is indirectly delivered from some other place. Needless to say, this was not entirely welcomed by his scientific colleagues. This seemed like a backward step. A neuroscientist, as Freud was at that time, arguing that dreams convey hidden meaning was a bit of an embarrassment. All the more so when you think of the method that Freud used to reach this conclusion. I'm sure most of you know, so I won't belabor the point, but the method was basically each element of the dream, as the dreamer remembers it, they must take each scene in the dream and they must say everything that comes into their mind in connection with that part of the dream. Freud said if you do that, this free association method, if you allow the dreamer to wander through all the thoughts that come to their mind in connection with the different elements of the dream, you find that they come together in a surprising or unexpected way. That all of those trains of thought join up and reveal another story, not the apparently meaningless nonsense of the dream itself, but rather a sensible thought, a coherent thought process that Freud inferred from these associations, that he said this coherent thought process lies behind the dream. What's more, Freud said this thought process is not only coherent and meaningful, it is always of the same type. The thought behind the dream is always in the nature of a wish. Not just any old wish, but a heartfelt wish. Something in the nature of a urge, a desire, something broadly speaking lustful and primitive. Freud said behind every dream is some sort of biological or some sort of animalistic desire, normally hidden from view, which emerges during sleep and then in a disguised form, in an indirect form, expresses itself in an attempt to fulfill that desire, in an hallucinatory attempt to fulfill that desire, disguised and distorted, and that is the remembered dream. Now, Freud said, if this is how the mind makes dreams, then how does the mind work? What can we infer from these conclusions, what the structure of the mind as a whole is? And he then set out his first model of the mind as a whole in his famous book on dreams, Die Traumdeutung, the interpretation of dreams. I'm emphasizing all of this, especially this last part of what I'm saying, in order to remind you that Freud's model of the mind as a whole was deeply rooted in his research on dreams. Freud said if dreams are a disguised and distorted conscious representation of something that lies beneath of a much more primitive, wishful, libidinal nature, then if this is the case for dreams, what can we infer about the mind as a whole? And as you know, he came up with a model that the mind as a whole works something like this. That behind the scenes of our conscious waking thought, our rational, sensible, reality-constrained cognition. Behind that, within us, there is an instinctual, affective, primitive, omnipotent, wishful mode of thinking. These two layers of the mind, Freud called ego and id. The ego represents the outside world, builds a model of the outside world, and tries to find ways within this outside world, through experience and learning, of meeting these deeper, primitive, ultimately bodily rooted needs of the organism. According to Freud, when we go to sleep, the ego, the part that represents the outside world, that's what goes to sleep. That's what sleep is. But the id, the part of the mind that is monitoring and representing the vital needs of the body, it can't go to sleep. If the part of the mind which represents your visceral homeostatic economy, the internal bodily economy, were to go to sleep, you die. So Freud, these are all the conclusions he came to. The conclusion was that because the ego goes to sleep, the part of the mind that normally inhibits the id is relatively, there's a shift in the balance of power during sleep, and the id emerges in a relatively unconstrained form. This, and here's an important part of the theory, this poses a threat to sleep. Because if your desire, your biological needs and urges are now released from inhibition during sleep, well, what are you going to do about them? You're going to act on them, more so than ever. And so Freud says this is why we dream. Because these the vital needs and primitive urges and desires are released from inhibition during sleep, they need somehow to be deflected from action, from the normal way we would, because that would undermine the need to sleep. So we have an hallucinatory, delusional fulfillment of our wishes. That, according to Freud, is what a dream is. A dream is an attempt to fulfill a normally repressed wish, a wish that would be repressed during waking life. It's an attempt to fulfill it in an hallucinatory fashion in order to stay asleep, because we need to sleep. The ego needs to rest and repair itself and take stock of the day's events. And Freud says the reason that these primitive wishes do not come out in raw form, that you don't see them in all of their brutish baseness, is because if we were to really consciously hallucinate our most heartfelt repressed desires, that too would be a threat to sleep. We would be shocked and horrified by our own actions. And this is why the sleeping ego, in a rough and ready fashion, does the best it can to camouflage what's going on. Now, I don't know how many of you in this audience are psychoanalysts or psychoanalytically minded people, as opposed to neuroscientists or just generally scientifically minded people. But the truth of the matter is, when Freud articulated this theory on the basis of that method, there weren't that many people who were persuaded. You know, it's a little bit far-fetched. It's a little bit lots and lots of theory on the basis of little bits and pieces of evidence, on the basis of a method which, as I've said already, as I've explained to you already, was not exactly experimental. It didn't exactly meet the rigors of laboratory science. Said Freud, well, you know, what can you do? It's in the nature of dreams, that they're subjective experiences, blah, blah, blah. But still, you know, it's a stretch. So, if you're mostly psychoanalysts, I'm sorry to tell you, it's a stretch. If you're mostly neuroscientists or scientists of any kind, I'm sure you'll agree that's a stretch. Nevertheless, Freud, a small group of people were persuaded by Freud, and they also applied this method of free association, and they too were persuaded that behind the dream, if you use the method properly, you'll find a a second, what Freud called the latent dream, as opposed to the manifest dream. You'll find an underlying thought process of the kind that I've mentioned, and this is very valuable because it gives you access to the deepest desires of the dreamer in a way that you don't have access during waking life. That group of people slowly grew. They're called psychoanalysts, and as it happens, they're meeting here in Prague as we speak. The International Psychoanalytical Association, those are the analysts. They're not the majority. The majority of Freud's neuroscientific colleagues in the 19th century, early 20th century were not persuaded. And I'll come back to that story just now. In psychiatry, psychoanalysis gradually acquired more and more prominence until by the 1950s and 60s, it really was the dominant frame of reference, especially in America, which at that stage was the dominant power anyway in the world. So American psychiatry and world psychiatry was strongly shaped by Freudian psychoanalysis, which, as I said, was in turn had its origins in a theory of dream and of why we dream. Considering the scientific foundations of the dream theory, it was perhaps a little not difficult to understand why what I'm going to describe to you happened next in the 1950s, why this was received with such excitement. But before I tell you what happened in the 50s, I want to tell you this. There was a philosopher of science, Karl Popper, who I'm sure just about all of you will have heard of. And Karl Popper, for the most part, is the accepted authority among my neuroscientific colleagues as to what counts as science and what does not. Popper, in his famous Conjecture and Refutations, which was published in the 1960s, said that a theory that is scientific has to have certain attributes. Anyone can come up with a theory of anything, but that doesn't make it science. If your theory is going to be a scientific theory, it has to give rise to predictions which can be tested and either confirmed or not. In other words, you say, I believe X, that's my theory. If X is right, then when Y happens, when I do Y, Z will be the result because of X. I believe X, therefore if I do Y, Z should result. And it has to be done, the experiment has to be done in such a way that Z might not result if your theory is wrong, and if Z does not result, then your theory is wrong. That's a refutation. If you can't do such an experiment, a critical experiment, where you test your theory by either confirming or not confirming a prediction from that theory, then it's not a scientific theory. That's the crux of what Popper claimed, and that positive idea has, as I said, come to dominate all of modern science. We all really think about Popper, which is no, it's not science if you can't test it. If you can't do an experiment which either leads to the result that you predicted or does not, then it's just, I think. God exists. That's my theory. Now, how do you prove that? How do you test that? What's the experiment? Well, I'll kill myself, and then we'll see if you can't do an experiment like that, so that's not a scientific theory. Everyone knows the theory that God exists is not a scientific theory. But what about the theory that dreams are an attempt to fulfill wishes in order to keep us asleep? Popper, for those of you who are analysts, I'm sorry to remind you, Popper used that theory as an example of what he calls pseudoscience. He said that something that's dressed up like science, that looks like science, it's Professor Freud, the neurologist who's saying this, and he says, I had a method, and if you follow my method, you come to this conclusion. It's presented as science. Popper said it looks like science, but it's not science because it doesn't make a prediction that you can falsify. Therefore, it's not science. For example, what would you predict if dreams are attempts to fulfill these heartfelt desires? You would predict dreams are delicious things. Your dreams are wonderful, experiences of your deepest lustful wants being satisfied in a hallucinatory fashion all night long. of course, as you know from your dreams, that isn't what they like. In fact, most dreams are pretty grim compared to what you would predict from the theory. But then Freud says, no, but that's not what you would predict from the theory. The theory predicts only that the thought that lies behind the dream is of that kind. And you have to interpret the censorship and the disguise, you have to get around it to find that hidden thing. And Popper says, you mean every time you can find that, no matter what that happens in the dream? And Freud says, yes. And then he says, well, then it's not falsifiable. And that's the whole point. It's this interpretation business. If you do it my way, you will find that this other thought lies behind it always. Popper says that's not science because you can't falsify the theory. So against that background, it was a great leap forwards when in the 1950s in Chicago, two physiologists named Azarinsky and Kleitman discovered a physiological phenomenon that occurs during sleep every night to all of us. Every night we go through this cycle from wakefulness down into deep sleep, our brain activity getting lower and lower and lower in its level of activation, until roughly 90 minutes into sleep, we have a period of activation, of arousal. Then we go back into deep sleep, and then roughly 90 minutes later, activation again and again and again and again throughout the night. These periods of activation of the brain during sleep were a real surprise. Because if you think about it, what's the brain doing being activated during sleep? Sleep is the very opposite of activation. And it's not just the brain that's activated. Other things happen too. While the brain is activated like that, your eyes dart around. Rapid saccadic movements of the eyes. Your heart rate changes. Your breathing rate changes. Your body temperature changes. If you're a boy, your penis changes. You get a whopping great erection. Very reliably every 90 minutes. So you are, in every sense of the word, if you're a woman, you also, your genitals are also engorged. It's just not so easy to see. You are in every way aroused during sleep, every 90 minutes, every night of your life. Azarinsky and Kleitman accordingly called this paradoxical sleep. The paradox being, how can you be so aroused physiologically while also physiologically in a state of quiescence called sleep? This led somebody in their laboratory to make a theory. This man's name, unfortunately, was William Dement. And Dement thought that when the brain is paradoxically aroused during sleep, this might be when the mind is paradoxically aroused during sleep. There is this paradox in mental life too, that you're unconscious and asleep, and yet suddenly you're conscious, and in fact very conscious, and you're having heartfelt, emotional, dramatic experiences. So Dement hypothesized that the state of rapid eye movement or paradoxical sleep is the physiological equivalent of the mental state called dreaming. Now, is that a scientific theory? Yep, because there's a prediction that comes from it. The prediction is that if you wake people up from REM sleep, they will be dreaming. And if you wake them up from non-REM sleep, from deep sleep, they won't be dreaming. Dement then set about doing that, testing his hypothesis, seeing whether his prediction came true or not. Now please note, he could either be right or he could be wrong, and that experiment will demonstrate it. If people dreamt the same amount during REM and non-REM sleep, then the prediction is wrong, which means the theory is wrong. The theory that REM sleep is the physiological equivalent of the mental state of dreaming would be falsified. So Dement did the experiment in the mid-1950s, and he found exactly what he predicted. That roughly 90% of awakenings from REM sleep lead to dream reports, and roughly 10% or even less of awakenings from non-REM sleep, from deep sleep, lead to dream reports. 90 versus 10. Not 100 versus 0, but they're asleep, for heaven's sake. You know, you've got to wake them up, and you've got to ask them to give a first-person report of what were they thinking about. And as you all know, dreams are notoriously difficult to remember. So 90 to 10 is, you know, as close as you need to confirm the hypothesis. And so from that time onwards, the vast majority of neuroscientists and sleep scientists have concluded that REM sleep is the physiological marker, the biological marker of the dream state. Now that was good news, because remember what I said about the scientific status of Freud's dream theory and the methods on which it rested, but also remember what I said about how influential it was at that time. As psychoanalysis completely dominated American psychiatry. You couldn't become a tenured professor of psychiatry in America at any decent university if you weren't an analyst. It was considered, you know, synonymous with academic psychiatry. Now, with the discovery of Dement and Azarinsky and Kleitman, we had an objective state that we could study using normal science, and we could discover what the brain mechanisms were that underlay dreaming, and we would begin to, in a more objective and less awkward and difficult methodological fashion, start to ascertain whether the sorts of things that Freud was claiming on the basis of his much less secure psychological methods and evidence, whether we could confirm from a physiological, neurological point of view that the dreaming brain worked like that. It was also a very fortunate fact that it's not only human beings who have rapid eye movement sleep in this cyclical fashion, all mammals do. So it was possible to use the tried and tested method of behavioral neuroscience on other creatures other than ourselves, to discover what the brain mechanisms of dreaming were. The leading scientist at the time was Michel Jouvet. And what Jouvet did was not to put too fine a point on it. What Jouvet did was progressively destroy different parts of the brain to see which bit was necessary for REM sleep. Obviously, you can't do that in humans, which is why it was happy for science that all mammals have REM sleep. And Jouvet took cats. Cat brain looks, this is a human brain, but Jouvet studied cats, whose brains look very similar to ours. And they also sleep a lot. And he made slices down the neuraxis of the cat brain like this, going deeper and deeper to see at what point did rapid eye movements stop during sleep. And eventually he made a cut over here, removing the entire forebrain, the whole of the organ of the mind, separating it from the brain stem. And still it had no impact on REM sleep. The cat still had the same cyclical alternating pattern. Jouvet cut deeper and deeper and deeper until he was here. This is the absolute primitive brainstem. Now here's the spinal cord, here's the medulla oblongata, and here's the pons. This is really basic. And it was only once Jouvet got down to this level, in the middle of the pons, that REM sleep stopped. What that meant was that REM sleep, that is to say dreaming, because remember, this is important, on the basis of Dement's findings, the conclusion was that dreaming and REM sleep are the same thing. When you look at the body from the outside, you see REM sleep. That's the physiological, objective manifestation of the subjective, psychological state that you experience if you are that body, called dreaming. Dreaming and REM sleep are identical. It just depends on whether you're looking from the outside or the inside. So what Jouvet was compelled to conclude was that dreaming is generated, it's caused by mechanisms at that level of the brain which are not mental at all. At that level of the brain, you're not thinking, you're not wishing, you're not having an Oedipus complex. You are controlling your heartbeat and your breathing and your eye movements and very, very basic things like that. Your muscle tone. All of the things which make up the state of REM sleep. The only mental function that's performed at this level of the brain is switching on the lights. Basic, just sort of like volume control, switching on consciousness. Nothing to do with the actual contents, with the actual workings of the mind. It's just level of consciousness. A student of Jouvet went further and found the precise mechanisms within that part of the brain that switch REM sleep on and switch it off. I don't want to go into too much detail, but basically there are cells in this part of the pons called the mesopontine tegmentum, which release a neurotransmitter in the forebrain called acetylcholine. So we call these cholinergic cells. They release acetylcholine in the forebrain, they activate the forebrain. There are other cells here in the dorsal raphe and in the nucleus locus coeruleus, here and here, which release other neurotransmitters in the forebrain, specifically serotonin and noradrenaline. And what Hobson, a student of Jouvet, was able to show was that when you go into REM sleep, these cells fire rapidly and these ones stop firing. And when you go out of REM sleep, these ones start firing rapidly and these ones stop firing. So it's on, off, on, off. Simple little switch. Hobson concluded that dreaming was an entirely basic physiological state. It had nothing to do with the mind. He said this explicitly, that dreams are not mental events, in the sense that they are not caused by anything mental. They are caused by an absolutely automatic, pre-programmed physiological switch, which every 90 minutes activates your brain, and then deactivates it, and then activates it, and then deactivates it. The cause is literally mindless. The way that it activates the forebrain is inherently meaningless, and Hobson also made this point very clear. It is not, there is no meaning to the activation. It is simply switching on the lights. You know, you switch on these lights and you see who of us are in the room. We might mean something, but the switching on of the lights doesn't mean anything. It's just... And so Hobson said, in a theory that he called the activation synthesis theory, he said that the mesopontine tegmentum, these meaningless, mindless cells here, activate the forebrain. The forebrain, which is the seat of the mind, has in its neural architecture thoughts, feelings, perceptions, memories, volitions, all the stuff of the mind. These representations are randomly, meaninglessly, mindlessly switched on by the chaotic signals coming up from the brainstem. The poor forebrain, of course, it generates an experience, it has to. It sees things, it hears things, it smells things, it thinks things, it remembers things, it feels things, it wants things, and so on. But these things are not meaningful. They're not generated by a mental state. They are just randomly switched on. And the forebrain then has to connect them all together and turn them into an experience, because that's what the forebrain does. And that experience is a dream. Hobson says, and this is also very important, he says, of course you can then go and look at the dream and find meaning in it. But it's you who's finding meaning in it. It's not in the dream itself. He said it's like an ink blot. You can make a random splotch of ink, and then you can look at it and say, oh, it looks like a butterfly, or it looks like a bat, or it looks like my mother's genitals or something. But that's in your head, it's not in the ink blot. That was Hobson's conclusion about dreams. Now, I've told you what the evidence for it was, and Hobson presented that evidence to a meeting of the American Psychiatric Association in 1976. And after presenting his findings, he took a vote from the assembled membership of the American Psychiatric Association as to whether or not Freudian dream theory still had any scientific validity in light of these findings. Now remember, this is the American Psychiatric Association. That's the center of power of world psychoanalysis at that time. It was no small fact, therefore, that the audience voted 2-1 against Freud for Hobson, and said, in light of these findings, we have to agree there is no longer any scientific validity in the Freudian theory. It's not possible that dreams are performing all these mental gymnastics and have all of these deep emotional and motivational meanings if these are the brain mechanisms that generate them. So it was against that background that I conducted a study, started in the middle of the 1980s, which took me a long time. I only published it in the middle of the 1990s, the first findings. That's because the study that I did not involve cats. It involved people. I wanted to see what happened when different parts of the brain are damaged. What is the effect of this on dreaming? Not on REM sleep, but on dreaming. And for that I needed human beings. Because you're not allowed to just remove little pieces of your experimental subjects' brains to see what happens to their dreams. If they're humans, somehow you are allowed to if they're not. But if they're humans, you're not allowed to. And so I had to wait for God to remove pieces of people's brains with strokes and tumors and infectious processes and the like. And if you take your time, eventually you can get exactly the same collection that you would want to produce experimentally, produced by nature. And by the mid-1990s, I was able to report what I had found in terms of what happens on the average when different parts of the human brain are damaged. What happens to their dreams. This is the basic method, as I said earlier, the basic method of behavioral neuroscience. If you want to see how a particular function is arranged in the brain, you have a look at what happens to that function with damage to different parts of the brain. It's the most basic method. Why I was doing it was because by then, by the mid-1980s, leading into the 1990s, it wasn't enough to say, as Hobson was, that the forebrain synthesizes, it joins the dots. The forebrain is a beautiful and complicated thing, different parts of which do very specific, precise mental operations. They don't just synthesize. So I was assuming that if the forebrain does what Hobson said it does, then damage to different parts of the forebrain must affect different aspects of dreaming. So that, for example, the visual part of the forebrain, if that was damaged, you might have non-visual dreams. If the auditory part is damaged, you might have dreams without hearing anything. Or if Broca's area was damaged, you might have dreams in which you don't speak. Or if the motor strip is damaged, you might have dreams in which you're paralyzed. Or if the frontal lobes are damaged, you might have even more nonsensical dreams than ever, because this is the part of the brain that organizes our rational thought processes and so on. That's what I expected to find. This is a brief summary, which I won't go into all the details, of what I did find. In 360-something patients, I found that these areas colored in here had predictable, consistent effects on dreaming. I'm going to mention only 4 of them. The first is this dark blue area here, which is the dorsolateral prefrontal lobes. It's this area. Remember I said that my prediction was that since we know that part of the brain is our rational, goal-directed, problem-solving, sort of civilized, inhibitory regulator. It's the executive ego, if you want. I thought that damage there would lead to even more irrational and random and chaotic dreams than we normally have. Well, what I found was the opposite. Damage to that part of the brain does nothing to dreams. If you take the dreams of patients with no prefrontal cortex, even though they are terribly disabled in their waking life, they really are in a lot of trouble. They can't think properly, they can't control their thoughts, they can't inhibit their impulses, they can't reflect on their actions, they don't obey basic social rules. They're like little kids. despite the fact that this is what happens to their waking cognition, their dreams are completely the same as yours and mine. Raters can't tell the difference between them. So that's the first finding. The second finding is much more interesting. That is that damage there, in the pons, damage here, in the part of the brain that generates REM sleep, has no effect on dreaming. My prediction was these patients will no longer dream. But they do still dream. In fact, when I say no effect, it's not true. They do dream less frequently, but they still dream. And this really puzzled me. Each one of these patients that I got, one after the other, they all continued to report dreams. And that was just simply a shock to me. Eventually, I had 18 patients with damage to the part of the brain that generates REM sleep, and yet they continued to dream. So, being young and insecure, I went back to the literature to find, I knew I must have got this wrong, because we'd known since the 1950s that part of the brain generates REM sleep, and that REM sleep is the same as dreaming, and yet all of my patients were dreaming. So I went back to the literature and found that since the 1950s, over 40 years of research, it had been demonstrated on many occasions that damage to that part of the brain in humans, just as in cats and rats and all other mammals, damage to that part of the brain in humans leads to loss of REM sleep. It had never, not once, been demonstrated, not even in a single case, that damage to that part of the brain leads to a loss of dreaming. That was really a surprise. How could we, as a discipline, sleep science, for 40 years have asserted that this part of the brain generates dreams without ever testing that hypothesis? I think the reason that we never tested it was because consciousness or subjective experience is an embarrassment to science. Subjective experience is just that, it's subjective, and science seeks objectivity. So if you have a subjective thing called a dream, and it correlates with an objective thing called REM sleep, once you've established that correlation, bye-bye dreams, You don't want to study the fluffy stuff of the subject anymore. You've got the objective stuff of the body, and you study that. I think that's why we didn't bother, once we had established that correlation, we didn't bother any longer with brain mechanisms of dreaming. Dreaming being this thing that's so difficult to study, we just studied the brain mechanisms of REM sleep in other animals, in whom, of course, there's no question of talking to them. You can't ask a rat whether, on losing REM sleep, whether it's also lost dreams, which is what you would predict. But even in the human subjects, and I must emphasize this again, even in the human subjects, many of which had been reported in the literature, in most of them, the investigator didn't even ask about the dreams. They just demonstrated that REM sleep was lost. But in a few cases, they did ask, and in a few cases, the patients themselves reported preservation of dreams. So, on seeing this, I concluded that my 18 patients meant what I thought they meant, that dreaming and REM sleep are not the same thing. It's a basic methodological error, because two things correlate with each other doesn't mean they're the same thing. Babies cry when they're hungry. That doesn't mean that if you want to find the mechanism of crying, you must look in the digestive tract. And that's exactly what we have done with dreaming. Dreaming happens during REM sleep. REM sleep is generated in the pons. That doesn't mean that dreaming is. It's not. It actually can't be. It's not possible that if you can have damage to the part of the brain that generates REM sleep and dreaming is conserved, dreaming cannot be the same as REM sleep. And in fact, what I found next proved that even more. These parts of the brain marked at the back over here are visual and spatial parts of the brain. And it's damage to the inferior parietal lobule and parietal occipital mechanisms around here. This part of the brain, when it's damaged, patients do stop dreaming. That's not a surprise. They can't generate normal visuospatial cognition. They can't generate mental imagery normally. So the basic cognitive processes that are required for dreaming are lost in these patients, and so dreaming is lost in these patients. But REM sleep is preserved. So what that means is we have what we call a double dissociation. A double dissociation, if there's damage to this area, then there's loss of REM sleep and preservation of dreaming. Damage to this area, there's loss of dreaming and preservation of REM sleep. That means dreaming and REM sleep are doubly dissociable. That means they're not the same thing. Absolutely, definitely. Cannot possibly be the same thing. So that's interesting. But the most interesting part of my findings that I published in the mid to late 1990s is marked in red over here. Red means important. These areas here, if they're damaged on both sides of the brain simultaneously, this is the white matter of the medial frontal lobes, the ventromedial frontal white matter. If that part of the brain is damaged, this part here, not the cortex, but the white matter underneath this part, if it's damaged on both sides of the brain simultaneously, you also lose dreaming. Now, the fact that damage here in the visual and spatial parts of the brain led to a loss of dreaming was not surprising. But the fact that damage in this red area led to loss of dreaming was really a big surprise. I couldn't understand it. Now, I told you I had 365, I think it was, maybe it was 361 cases. Whatever it was, of those, I only had 9 patients with this damage marked in the red area here. And 9 patients is not enough, but they existed. And it was, in fact, the first time that I had a patient like that who reported loss of dreaming, I was annoyed because it wasn't in the area that everyone else had the damage, so I thought it was undermining my findings. And then another one, and then another one, you know. And then I thought, oh, it's all going wrong, it's all going wrong. Until I realized that these patients all had damage in the same area, that there was 2 areas of the brain that were necessary for generating dreams. One, the visuospatial part, and another, the God knows what part. It's not at all clear what's happening here that's so essential for dreaming. And this part of the brain has no obvious connection with the dreaming cognition. So that then became the main focus of my research from the late 1990s onwards. I wanted to find what is it that's going on in this part of the brain that's so essential for dreams. Now, in the white matter of the ventromedial frontal lobes, there are connections between everything and everything. So it's really trying to find a needle in the haystack to try to find what part of this is the crucial part for dreaming. Hobson's view was that it was cholinergic fibers. I told you that these are cholinergic cells here. They send messages to the thalamus, which in turn activates the forebrain. When Hobson saw that I'd found that damage to this part of the brain leads to a loss of dreaming, then he said, well, there are also cells over here in the basal forebrain that send cholinergic fibers through this area. Now that's true. There are cholinergic cells there. But isn't it interesting that they've got nothing to do with these pathways? Now he was wanting to claim that it was these cholinergic pathways that generated dreams. And in any event, I only had 9 patients. So I looked at the scans in my 9 patients. Here they are. And they reminded me of something. I don't know if any of you are old enough like me to be reminded of something when you look at a scan like that. What it reminded me of was this. There was an operation in the olden days that used to be performed on patients who had hallucinations and delusions. Positive psychotic symptoms were well controlled by this operation, which cut the fibers of the ventromedial frontal white matter. The operation was done on hundreds, in fact on thousands of patients. So So since I only had 9, I once again went back to the literature to see had any of these thousands of patients said anything about their dreams. Since that operation was done in the 40s and the 50s and the 60s, when psychoanalysis dominated psychiatry, I thought perhaps the people who did the operation would ask their patients about their dreams. And indeed they did. If you go back to the large series of cases, hundreds of cases reported by, for example, Jerome Frank in America, 500 cases of this operation, which was called prefrontal leucotomy. 500 cases. He observed the following. The operation leads to a reduction, a great reduction in hallucinations and delusions. Secondly, it results in a dramatic loss of motivation. The patients become aspontaneous, avolitional, inert, apathetic, akinetic. Thirdly, it leads to a loss of dreaming. Frank reported this in the 1950s. Partridge in England reported the same thing, exactly the same thing, again in hundreds of cases. And Schindler in Germany reported the same thing, in hundreds of cases. The operation leads to loss of positive psychotic symptoms, loss of motivational drive, and loss of dreaming. Schindler went so far as to say that if the patient, after the operation, continues to dream, it's a bad prognostic sign. And if you think about it, it makes sense. Dreams are hallucinations and delusions. So if you can still produce dreams, you can still produce hallucinations and delusions. So it's a bad sign. And that got me thinking about what part of that spaghetti junction, all of those fibers connecting up with others, what might be the important part for dreams. Because coursing through this area, as I said, are fibers connecting everything to everything. But specifically, there's this fiber pathway, which starts in the ventral tegmental area here. This circle should be there, it fell down. This is the ventral tegmental area here. And it projects through this area and releases at its terminals a neurotransmitter called dopamine. Now, the reason that this operation stopped being done was not because of public outcry. It was because it was replaced by a drug. The drugs which were introduced in the 50s were first called major tranquilizers, that is antipsychotics, which have the basic action of blocking dopamine. They block activity in this pathway. So this pathway, which is severed by the operation, is blocked by antipsychotic drugs. Blocking of dopamine. So that led me to the hypothesis that what is leading to the loss of dreaming in these patients is the same thing that's leading to the loss of the ability to produce hallucinations and delusions, that is, loss of dopamine due to severance of this pathway. Now, I told you that Hobson had an alternative theory that it was severance of cholinergic pathways in that same area. And here again you have the beauty of the basic scientific paradigm of Popper. My prediction would be block dopamine, you have loss of dreams. Increase dopamine, you'll have more dreams. Hobson's prediction would be block acetylcholine, you'll have less dreams. Increase acetylcholine, you'll have more dreams. Lovely. Two competing predictions. Can't both be right. So, what do we find? If you block dopamine, you greatly reduce dreams. They also become much more bland, much less emotionally intense, much less bizarre, and far fewer nightmares. You increase dopamine, you have more dreams, you have longer dreams, you have more emotionally intense dreams, you have more bizarre dreams, and you have more nightmares. With acetylcholine, exactly the opposite happens. If you block acetylcholine, anticholinergics, you get more dreams. In fact, you even get dreams while you're awake. Those of you who know anything about anticholinergics will know that scopolamine, for example, it will increase dreams. So the prediction, the exact opposite of the prediction, is what we find. Now, I must very quickly summarize other evidence. So we know that cutting this pathway leads to a loss of dreams. We know that blocking activity in this pathway leads to a loss of dreams. We know that increasing activity in this pathway leads to an increase in dreams. We also know, if you do functional neuroimaging, that that area is massively activated. By the way, it projects here to the nucleus accumbens. That's the main terminal point of this pathway. And the nucleus accumbens is absolutely switched on like a Christmas tree during dreaming sleep. By the way, this is a very nice image to look at. You can see here how the prefrontal lobes are not activated during dreaming sleep. That's exactly the same as we found with the lesion studies. But this emotional, motivational limbic area is massively activated during dreaming sleep. And so is the visuospatial cortex here, as the lesion findings showed. But the really telling ones were these two studies. The Dahan et al. Study of 2007 and the Lehner et al. Study of 2005. What those two studies showed was, the first of them showed that the cells of the ventral tegmental area, as we go into REM sleep, to dreaming sleep, remember that dreaming sleep correlates, dreaming and REM correlates. So during REM, 90% of the time you're dreaming. Those cells, they fire like hell. Like this. Not it's which is even better, because that's burst firing. Burst firing maximizes the release of the neurotransmitter at the terminal. And what Lehner et al. Found was, by directly measuring the neurotransmitter at the terminal, so these were single cell recordings, these were microdialysis studies, they found that there is maximum release of dopamine in the nucleus accumbens during dreaming sleep, even more than when you're awake. Now, what's important about that, I need to just quickly emphasize, this is important. This system, it's called the mesocortical mesolimbic dopamine system. It's a sort of semi-anatomical, semi-physiological term. It's also, in behavioral terms, it's called the reward system, or the seeking system, or the wanting system. These are different names for it. Berridge calls it the wanting system, Panksepp calls it the seeking system, Rawls calls it the reward system. What all of those words have in common is positive motivation. It's a desire system. Nobody would doubt that. It's the brain's, it's the mammal brain's desire system. If you activate this system, what the animal does, and by the way, what activates this system is need. There's medial hypothalamic need detector mechanisms monitoring the bodily economy, the homeostatic economy of the internal body. When there are imbalances, when there are needs, they register in those nuclei which activate this seeking system. And what that makes the animal do is go out into the world and seek. Because it wants, it's in need. If you have a need of a biological kind, the only thing to do is to go out into the world, because that's the only place your needs are going to be satisfied. I'll make a little aside here. That's why life is difficult. Narcissism doesn't work. You can't just say, oh, I wish I had this, okay, I've got it. You can't. Especially when it comes to vital needs and sexual needs. Reproductive success and survival require you to go out into the world and get what you need. What you need is an object, and objects are out there. And that's why this system evolved. Whatever it is you need, mate, it's out there. Go forth and multiply. So if you want to know what this system does, think about you release a dog into an open field. Think about it. Your new puppy, and you put him or her down in an open field. What does it do? Well, it starts sniffing the air, twitching its whiskers, looking interested and optimistic and exploring. What's there? Oh, yeah, that's interesting. Whoops. Let me piss there. Oh, yeah, there's something to eat. Let me eat that. Oh, I'll drink that. That's what they do. It's just like, wow. The world is interesting. The world has got good things in it. I want to find them. It's foraging. Foraging is the word for it. And in human beings, it's called cruising. You drive your car, checking out the sea. Who's there? If you want to know what this system does, then, well, I wouldn't actually recommend this, but if you've ever seen anybody who's been so naughty as to snort cocaine,